In this lesson, we are talking about the process of translocation. But before we do that, it's better that we review the structure of phloem first, right? Though we have already done that in the previous lessons, so you can have a look at those videos also. Otherwise, definitely we are actually discussing it here as well. So when we talk about the phloem actually consists of these as actually sieve tube elements. So we have these cells here. Then you can see these are vertically connected to each other, right? So we have sieve tube elements and the companion cells, right? So these together they form the phloem, right? So we have these unique tube-like structures as you're looking at. So these are actually known as sieve tubes. This whole thing, the sieve tube. And then you can see one sieve tube. If you look, this is actually made up of many elongated cells. So you can see this is one cell here, this is another cell here, and these cells, these elongated cells, are actually what we call a sieve tube element. Okay, so a sieve tube is made up of many sieve tube elements which are connected to each other vertically, right? So they make the continuous vertical column that you see here. Now, each sieve tube element that you're looking at, this is not a dead cell, unlike xylem. When we were talking about xylem, so xylem, you saw that there was no end plate. Okay, there was nothing here that was stopping the flow of water and minerals, right? So we had this continuous mass flow. But sieve tube ke andar, what is happening? We have these sieve plates, okay, and these actually have these like pores in them, right? So this is the difference between, as you see, xylem, and then you actually compare that with the phloem, right? So here in these end walls, you see the end walls are not completely like क्या कहना चाहिए, they are still there, okay? They are there, but they have pores in them. Okay? So each sieve tube element has at least one companion cell lying close to it. So you can see ये पूरा sieve tube है. Each sieve tube is made up of sieve tube or sieve elements. Every sieve element has a companion cell lying next to it. ठीक है, so यहाँ पे भी आपको companion cell नजर आ रहा है, इधर भी नजर आ रहा है, ठीक है. So these are all companion cells. Then you can see we have these lateral sieve tubes as well here, which actually connect the companion cell to the sieve tube element, right? So this is a basic structure, and remember, these are not dead cells. So here is another diagram, okay, just for the comparison. So if you look at this as this sieve tube, and then if you look at this one sieve element here, actually, so what we notice is here is that there's a sieve plate. So end plates, yeah, end walls are not actually completely destroyed. Okay, so we have these sieve plates which actually have these sieve pores in them. But what you also notice is that cytoplasm is actually pushed towards the periphery. Okay, it is not completely filled. Like if you actually compare that with the companion cell, so they are the complete cell that you would actually see in a typical plant cell, right? But here, the cytoplasm is pushed towards the periphery. Then you can see a very few organelles, like we can only see a mitochondria on here, endoplasmic reticulum. But besides that, you would not find any nucleus here. Tonoplast, definitely there's no vacuole here as well. So tonoplast or ribosome. So these structures are not found in the sieve elements, right? So no nucleus, no ribosome. So definitely nucleus is not there. So definitely no protein synthesis has to take place. So there's no need for ribosomes as well, right? But if you look at the companion cell, so it's complete cell. It has all the features that a cell would have, a plant cell would have. So you can see a lot of mitochondria there. And there's a reason as well, because again, we will see how the translocation actually is done through active transport as well. Okay, so you can see a lot of mitochondria there. We'll need ATP. You can see the endoplasmic reticulum, nucleus, vacuole. So you'll see all the features of a cell, right? And then you can see the companion cell, and these sieve elements are again connected to the plasmodesmata. Okay, so this is how, and this whole structure is actually what we call a phloem. Okay, so it's a phloem sieve tube element along with this companion cell. All right, then here we are looking at the actually what does this phloem sap contain, or what actually is being translocated across the plant. So there's a list of organic compounds as well, and inorganic minerals as well. But what you need to remember basically is sucrose and amino acids, right? These are the two that you need to focus on. And these are organic compounds that we also call as assimilates. Okay? The word assimilates is used for them. Assimilates are organic compounds, right? In this case, we have sucrose and amino acids. Then here actually what they've shown you is a comparison between a xylem sap and a phloem sap. Just for understanding, see xylem only the mass flow is there only of water and the mineral ions there, right? So you will definitely not find any sucrose there. That if you compare them with phloem, so sucrose, like the major component there is sucrose, right? And then look at the amino acids. So these are the two major components that you're going to find in the phloem cell, and these are the two basically that we'll talk about. Or paper, maybe if you're looking, you're writing down the answer, so make sure you use both of them, sucrose and amino acid. Or just please do not write sucrose only, amino acid only, दोनों को एक साथ लिखेंगे, you'll get the full credit then, right? Then you can look at the other minerals as well, okay? But please make sure you focus on these two. All right then, so we are talking about phloem. So phloem, generally what it does, it transports organic substances in the plants, right? And what it transports is, we have already discussed, it mainly transports the sucrose along with amino acids. So please make sure sucrose plus amino acid, it transports both of these around the plant. Okay? Then sieve tube elements, as you were looking at these here. So these are the sieve tube elements, okay? Elongated cells. Okay? These are living cells. Please make sure you remember that these are not dead cells. Okay? These are living cells, but they do not have any nucleus. They form the tube-like structures for the transport of solutes. And for each sieve tube element, you can see there are companion cells next to them. So this is a companion cell, and then you can see it has a nucleus, right? So companion cell with the nucleus is there. It will have all the cellular organelles, okay, that are found in a typical plant cell, right? And companion cells carry out all the metabolic activities for the sieve cells, right? So because they have all the components, and then you can see the plasmodesmata. Okay, so components will then be transferred to the sieve tube elements. All right, and this is another diagrammatic view, so just have a look, focus on the things, and then we'll move on to the next one. So just you can compare between the phloem sieve tube elements and the companion cells and see how they're connected. Okay, then let's talk about the process of translocation now. When we talk about translocation, please remember translocation simply means that you're literally moving something from one place to another. So it is not just associated with phloem, okay? It is actually transport. When we talk about transportation, so it can be both from xylem and phloem, right? But commonly, TKS is more commonly actually tends to be like described for phloem, okay? So phloem के लिए इसलिए हम translocation use करते हैं, but remember कि xylem में भी हो रही है. So it tends to be used more commonly to describe the transport of soluble organic substances within the plant. ठीक है? So soluble organic substances की क्योंकि हम बात कर रहे हैं, we are talking about sucrose, we are talking about amino acids, right? Or generally we are talking about these assimilates. ठीक है? So इसीलिए generally translocation का term वहाँ पे use हो रहा है. ठीक है? And translocation के लिए actually what organic substances are we talking about? The soluble organic substances are actually the substances which are made by the plant itself, like photosynthesis हुई, sugars बनी, ठीक है? And then like definitely those have to be transported then, ठीक है? So these materials have to be transported, right? And these organic compounds are known as assimilates as well. ठीक है? So translocation is the movement of nutrients around the plant. ठीक है? The term includes the movement of minerals, which can be dissolved in water and transported in the xylem. As we have just said, translocation does not only occur in phloem, okay, but usually refers to the transport of sugars, amino acids, and other organic molecules in the phloem. Okay, so make sure you understand the difference. Then it can occur in either direction in the phloem, so it is bidirectional. When we're talking about xylem, so xylem it was unidirectional. Okay, it was a passive transport. Phloem ke andar, when we're talking about translocation, so translocation happens in bidirectional. Okay, wherever the nutrient is required, wherever that organic compound is required, it will move. Okay, so agar roots pe zarurat hai, to it will definitely go down. Agar aage kisi jagah pe stem pe zarurat hai, it will definitely go there, right? And it is an active process. Okay, so unlike transpiration is a passive process, it is unidirectional, but translocation it is an active process. Okay, it requires energy. Okay, unlike the water transport in the xylem, right? So it's bidirectional and it requires active transport as well. All right then, as we're talking about the phloem translocation, so we need to actually talk about the transportation of a starch basically. And remember, जब हम starch की बात कर रहे हैं, तो remember it is sucrose, ठीक है, that is being transported through the sieve tubes of phloem tissue from the source end up to the sink end. So these are the two terms that you need to know. ठीक है, कि source actually is the place जहाँ से वो starch या sucrose actually is going to move. ठीक है, जहाँ पे उसकी storage हुई है. and sink is the place where it is actually going to be consumed or utilized. Okay, so you need to know what is the source end and what is the sink end. Okay, and remember these are not fixed, right? So this occurs in both directions. The two ends of the phloem translocation are as follows. So source end and the sink end. ठीक है, source end की जब हम बात करते हैं generally जहन में आ सकता है कि yes it can be the leaf जहाँ photosynthesis हो रही है, but remember it cannot always be the case. ठीक है, now for example if it's a tuber, ठीक है, और starch की form में वहाँ पे definitely store हुआ हुआ है, and when that starch aerobic cellular respiration के लिए you need glucose, the plant needs glucose basically, that the starch has to be broken down. ठीक है, starch broken down हो रहा है into maltose and maltose into glucose. That glucose is actually being used in the aerobic cellular respiration. तो उस केस में वहाँ वो जो storage site है starch की, that is actually the source end. ठीक है, so remember it is not always कि leaf will be the source end. ठीक है, so source end क्या है? It is the location in the plant where the sugar is being produced either by photosynthesis or by breaking down of storage. ठीक है, so remember it is either the site of production, which definitely is the leaf जब photosynthesis के through बन रहा है, or it can be the breaking down of the storage starch. So both of these can serve as the source end. ठीक है, sink end क्या है? It is a location in the plant where the sugar is being stored or consumed. ठीक है, store भी जब हो रहा है तो एक time होगा कि definitely अगर ज़्यादा sucrose है या glucose है, eventually it has to be stored somewhere. तो ठीक है, जब तक वो store हो रहा है, whatever you are storing, it actually is the sink end. ठीक है, or it is definitely consumed is the sugar sink. So remember source and a sink end, ठीक है, ये depending on the situation, ठीक है, will be different. All right then, so let's go to the examples of what can be a source, what can be a sink, right? So sources, when we talk about the sources, actually these provides assimilates, and assimilates as we talk about कि हमें दो चीजें याद रखनी है, sucrose plus amino acid. ठीक है, so these provides assimilates to the plant from reactions, ठीक है, or storage for transport. ठीक है, then so what can be the sources? So definitely green leaves and stems. ठीक है, आपको पता है green leaves में photosynthesis हो रही है, so definitely the site for the source end. ठीक है, as well as the storage organs. The storage organs क्या हो सकते हैं? The tubers, the root taps. ठीक है, या food stored in the seed. Seed के अंदर भी basically जब endosperm के अंदर starch की form में है, so that is actually the source. So sucrose makes up 20 to 30% of the phloem sap, right? If you look at the sink, the sink they use the assimilates from the plant in various processes. ठीक है, तो कौन कौन सी जगह है जहाँ पे different processes के लिए ये use हो रही होंगी sucrose? That can be the growing roots. Definitely growth के लिए they need energy, they need glucose definitely. ठीक है, so cellular respirations होंगी, metabolic reactions होंगे वहाँ पे. Active processes in the roots and the stems. Meristem cells definitely they are growing and dividing. Developing stores as well, like seeds, fruits or storage organs. जैसे जैसे fruit develop भी हो रहा है, तो eventually it has to grow to a certain size. So by the time it is growing, तो definitely is a sink at that time. वो nutrients को absorb करेगा, अपने अंदर store करेगा. ठीक है, तो जब तक वो complete fruit नहीं बनता, it is definitely a sink, right? So just remember that you know what are the sources, what are the sinks. All right then, another thing that you have to like understand here is actually the process of phloem loading and unloading basically. ठीक है, so as we are talking about yes, sucrose actually is transported, sucrose and amino acids. ठीक है, and right now we are talking about sucrose right now. So sucrose actually from the site of production or from the source end, it has to be loaded into the phloem, and then from phloem it will actually be transported to different parts of the plant wherever it is required. Okay, so as this sucrose is actually being sent into the phloem, so the process will be known as phloem loading. Okay, or similarly, this sucrose as it goes to the different parts of the plant, let's say it is going towards the root. Okay, wherever it is required. अब वहाँ से when this sucrose is actually going to go into the root, so तब हम उसके लिए term use करेंगे phloem unloading. Okay, so we use these terms, phloem loading and phloem unloading. So translocation, yes, we know is a vital process. Definitely. Plants, for survival, they need these nutrients, they need the assimilates, organic compounds, they need them, right? So if you talk about a large tree, it can move up to 250 kgs of sucrose around its trunk in a year. So this much sucrose is transported across around its trunk in a year, in a large tree we are talking about, right? So assimilates are moved into the phloem by active processes, right? So energy is required for that. So there are two routes. ठीक है, assimilates are moved into the phloem. ठीक है, so these are the symplast root and the apoplast root. So we have already talked about okay, which like what does Symplast root and what does Apoplast root mean? We have talked about these in the Xylem. We were studying Xylem as well, or actually transport of water की जब हम बात कर रहे थे. But remember, basically, apoplast is actually the movement across the cell membrane. Sorry, cell wall. Sorry about that. So the movement across the cell wall, ठीक है, or this movement of these organic compounds that assimilate through the cell wall. ठीक है, so that is apoplast, or Symplast क्या है? The movement of these assimilates through the cytoplasm. ठीक है, so these are two roots that can be used by the plants, ठीक है, for the purpose of translocation. Symplast root and the apoplast root, right? So when we talk about the Symplast root generally, most of it is a passive process. Okay? So one reason of that is, it is that yes, that assimilates are moving across the cytoplasm, but then one cells are connected to each other through plasmodesmata. Okay? So they can be transported from cytoplasm of one cell to the cytoplasm of the next. So assimilates, okay? Generally, they are actually stored in the vacuoles of the cell. Okay? And these assimilates are moved through the cytoplasm of the mesophyll cells into the sieve tubes across the connecting plasmodesmata. Okay, so if you look at this diagram here, okay, so you can see that these connecting plasmodesmata. So iske through when they're moving, okay, from cytoplasm to this, so ये एक कहलाएगा आपका Symplast pathway. Okay, so that is a Symplast pathway. So they move through the cytoplasm mesophyll cells into the sieve tubes across the connecting plasmodesmata. So this generally is a passive process. Assimilates are moved by changes in the water potential of the cells. Okay? So this is a Symplast pathway or Symplast route, and this is generally passive. Apoplast pathway, however, requires active transport. So again, one way you can remember is that apoplast A se shuru ho raha hai, so it involves active transport. Okay? So this is the difference between Symplast and apoplast pathway as well. What is happening in apoplast pathway? Assimilates diffuse through the cell wall and the intermembrane spaces. ठीक है, apoplast के through गुजर रहे हैं assimilates. ठीक है, अब what is happening? अब let's say कि there is a source and let's say यहाँ पे एक leaf है. ठीक है, the food was actually made in the leaf. ठीक है, now it has to be transported. But अब जो pathway होगा, तो from the source, first it will have to go into the companion cell. ठीक है, अब companion cell में when it is entering, so there we actually require the apoplast route definitely. So when these assimilates reach the companion cells, they are actively transported across the membrane into the sieve cell cytoplasm. Okay, how it is happening. Okay, so जब वो companion cells में जाते हैं, ठीक है, so they're actively transported across the membrane into the sieve cell cytoplasm. So let's see how it is happening. So you will have to focus on the companion cells at the moment here. ठीक है, remember when we were discussing the structure as well, we saw that these cells have a lot of mitochondria in them as well. ठीक है, so we need a lot of ATP, to active transport will definitely take place here. So let's see how is this happening. If you focus on this diagram here, see what is happening. See, this is the source, let's say a leaf cell, where photosynthesis is happened and definitely sucrose is formed. What is happening? Sucrose cannot enter the companion cell directly. Okay. So what is happening? Okay, one thing we noticed, एक चीज हो सकती है, we saw that Symplast pathway is also going to take place. Okay, which is largely passive process. कि चलो sucrose enters the plasmodesmata, then through the companion cell it enters the phloem sieve element. Okay, but right now we are talking about the apoplast pathway. So for the apoplast pathway, what happens is that we have these hydrogen pumps here along the cell wall, okay? Cell membrane, in fact, in the companion cell. So this hydrogen actually is pumped actively from the companion cell into its cell wall. Okay? So it is actually pumped out of the cell, right, into the cell wall. What is happening in the cell wall? The pH actually becomes low, and then the concentration of hydrogen ions actually increases here. What happens because of this? Because now there's a concentration gradient, there's more hydrogen ion there, so they will definitely enter the cell again. But what they do now they actually act as co-transporters, and they will bring in sucrose along with them, right? So right now we are talking about the apoplastic pathway. So the active hydrogen pumps, so which you're looking at here, they're driven by the ATP pumps. So hydrogen ions are pumped out of the companion cell into the cell wall. And remember, we're talking about the apoplastic pathway. Okay, so they will be pumped, they will definitely cross the cell surface membrane, they will come here into the cell wall. Okay, following the apoplastic pathway. What is happening after that? This creates a high concentration gradient of hydrogen ions in this wall. Now hydrogen ion sucrose. Now there is a pump here, okay, which this is a hydrogen ion sucrose pump, right? So as the hydrogen ions are moving inside the companion cells again, okay, so through this carrier protein, what will happen? They will bring along sucrose as well, okay. So this is what we call as like, so active transport actually happened here. This hydrogen was being sent from the companion cell to the cell wall, right? But then it enters as a code, enters like it through the carrier protein, okay? It enters the cell along with sucrose, right? So this is the apoplastic pathway, okay? And this is what we call as the Phenome loading. So sucrose, okay, now it has come into the companion cell. Now from the companion cell through the plasmodesmata, it can go into the phloem sieve element, right? So this is the pathway that is followed. Then here in this case, as sucrose enters, so definitely what is happening because of that, okay, now the solid potential is more in this case. What will happen? We definitely have xylem next to it. So what will happen because of that? Water will flow in because of osmosis, because now there's more solid potential. Water potential becomes low. So water flows in by osmosis, right? So because of that, what happens? This increases the pressure now. So pressure driven mass flow of water and solutes takes place then. Okay, so it can then flow, okay, towards the root if it is required there. Okay, so this is how it actually happens. So have a close look at the diagram one more time, try to understand it, and then we'll move on to the next part. This is another diagram that is showing you okay, how hydrogen is actually sent actively through this proton pump outside the cell. It creates a high proton gradient there, right? And because of that, it enters through the co-transporter, which is a carrier protein, but it brings along sucrose along with it as well. Okay, and then the sucrose from the companion cell can then go into the sieve tube element, okay, through the plasmodesmata. Okay, as sucrose actually enters, so this actually brings along water as well through osmosis, okay, and the pressure driven mass flow of water and solutes takes place. So this is the process of Phenome loading. Okay, this is another view. So here you can look at both the Apoplast pathway and the Symplast pathway. So if you follow the Symplast pathway, so this is the mesophyll cell. So you can see that sucrose definitely formed here, but it will move from, because Symplast pathway involves the movement of these assimilates to the cytoplasm. So they will move from the plasmodesmata of one cell to the next, and then you see okay, we have these phloem parenchyma, then comes the companion cells, and then comes the sieve tube element, right? So they will follow through the cytoplasm into the plasmodesmata, and they will actually load here. And if you look at the apoplast route, apoplast actually involves active transport. So see here, as the sucrose is actually being transported through this phloem parenchyma cell. So now you see, okay, because it has to follow the apoplast pathway. So in this case, now hydrogen will be sent out of the companion cell into the cell wall, and now it will enter the cell again through co-transporters, but bring in sucrose along with it as well, right? And after that, right, that sucrose can then again go inside to the plasmodesmata, right? Or it could follow this route, right? So this is how the apoplast role will actually be involved in active transport of sucrose in the phloem. So have a close look at the diagram, try to understand it. Okay, so the mechanism of phloem sap translocation. So let's review. So the starch, which is the main photosynthetic product, is transported in the form of sucrose as an aqueous solution, because sucrose is soluble and moderately inactive than glucose. This takes place through the sieve tubes at the source, and sucrose is actively loaded into the sieve tubes through the transfer cells. This is known as Phenome loading. This increases the Solute potential of the sieve tubes decreases the water potential. Then water enters the sieve tubes by endosmosis from the xylem vessels. This builds up a high hydrostatic pressure in the sieve tubes at the source end. At the storage organ, sucrose is actively unloaded into the storage tissues through the transverse cells. This is known as phloem unloading. Because of that, what is happening? The solute potential becomes less and the water potential becomes high. Right now we're talking what is happening at the sink end, right? So this lowers the solute potential, increases the water potential. Because of increased water potential, water will move out. It will leave the sieve tube by exosmosis and it will enter the xylem tissue. This builds up a low hydrostatic pressure in the sieve tube at the sink end. Okay? Due to this, a hydrostatic pressure gradient from the source end to the sink end along the sieve tubes is created, and then you know how it will flow from the high water potential to the area of low pressure, water potential gradient, right? Then the phloem sap is transported to the sink end up to the source end by mass flow. Okay? Because right now what is happening? Hydrostatic pressure अब नीचे ज़्यादा हो गया क्योंकि अब वहाँ से सॉल्यूट्स चले गए हैं अब सिंक में, which can be the root, let's say. Hydrostatic pressure ज़्यादा हो गया और ऊपर कम है. अब what will happen? This phloem sap is transported, ठीक है, to the sink end up to the source end by mass flow. ठीक है, this mechanism is known as pressure flow hypothesis. ठीक है. So due to this, hydrostatic pressure is actually generated from the source end to the sink end, right, along the sieve tubes, and then the phloem sap is transported to the sink end, and this actually is known as the pressure flow hypothesis. So here you can have a look at the diagram as well. How is this phloem loading and unloading taking place? So you can see this is the actually leaves where photosynthesis is taking place. So this is the source. So carbon dioxide plus water, okay, you get sucrose, water. But what is happening now? Now here the concentration is more. So eventually they will be loaded in the sieve tube through the companion cells, right? So here you can see the phloem loading, okay? Then definitely because of this, the solute potential would increase here. As the solute potential increases, water will flow in from the xylem. Okay, this will create a hydrostatic pressure. So water will, this sucrose or these assimilates will move in as a bulk flow. Okay, they will go down to the sink. Okay, then definitely at this place, they will be unloaded. Okay, as the sucrose is unloaded, so this decreases the solute potential here. Okay, and but what happens because of this, water potential becomes higher, right? And then as water potential becomes higher, so water can go to the xylem as well as it can follow sucrose here, because now the solute potential is more here. Now the sucrose, which is actually being unloaded, it can then convert into glucose, fructose, right? And then this glucose can then, if it has to be stored, it will convert into starch. If it has to be used by the plant cells, it will convert into cellulose. Or if it is used in aerobic cellular respiration, so it can definitely produce carbon dioxide and water, right? So this is the whole diagram that is showing you the sources, the sinks, and how mass flow is taking place in the phloem. So you can actually have a look at the diagram, pause the video. Okay, so if you're talking about the mass flow hypothesis, so this is just a recap, right? So in the mass flow hypothesis, we know there are two ends. We have the source end and we have the sink end. So if you look at this diagram, so they've shown you the source cell, which is the leaf, and this is the sink end, which is the root, right? So the loading of these solutes from the companion cells. So these are the companion cells. Okay, it will take place at the source end, right? So as they actually enter here, this will reduce the water potential inside the sieve tube. What will happen because of that? Since solute potential is more, water potential is less. So you can see water will actually move in from the xylem. Okay? And because of diffusion, and what will happen now, this solution will actually move towards the sink as a mass flow or as a bulk flow. And now these solutes will be unloaded, okay, from the sieve cells to the companion cells to get the sink. So you can see here. Then what will happen? Here in this case, what is happening then, water potential will definitely increase here because now the solutes have been unloaded. And now this water will actually through osmosis will go back to the xylem, right? And this is how this is actually the mass flow hypothesis. So this is another version. So you can actually use these for your notes. How do sugars move along the phloem? So see what is happening. Number one, sucrose is actively loaded in the sieve tube element, reduces the water potential. Water flows in by osmosis, increases the hydrostatic pressure in the sieve tube element. Then it moves down the sieve tube from high hydrostatic pressure to at the source to the low hydrostatic pressure at the sink. Then sucrose is removed from the sieve tube by surrounding cells, increases the water potential in the sieve tube, and then water moves out of the sieve tube, reduces hydrostatic pressure. Right, so you can use these points for your notes. Right. Now, since we are talking about these are hypotheses actually, right? So when we talk about these translocation or movement of these assimilates across the plant, so there are a few supporting evidences, okay, and definitely there are things which are objections, which are objecting this whole theory. So what are the evidences which are supporting, okay, the mass flow? So they're saying that when this ring of bark from a woody stem is removed, so remember the bark, because xylem is inside and phloem is outside, जो bark होती है tree की, that includes phloem. ठीक है? और phloem के लिए हम बात क्या करें कि it is actually the area where this translocation is taking place. So if you remove the bark, एक tree है उसमें से from a portion you actually remove a ring, ठीक है? So a bulge is formed above the ring. ठीक है, उस रिंग के ऊपर जहां से आपने रिंग रिमूव की, a bulge is formed there. So a high concentration of sugars is found within the fluid from the bulge. ठीक है, so below that ring, जहां से आपने ring पूरा remove किया है, तो above that ring, तो you will see a bulge which contains these the sugars there. The high concentration sugars will be there, but below the ring, ठीक है, this will not be the case. So this indicates a downward flow of sugars. So when we say from the source, जैसे leaves आपके source है, उसके through it is going down, or let's say it has to go towards the roots. So what will happen अगर आपने tree से एक bark remove कर दी, तो because it is traveling down, तो अब वो phloem वहां पे है ही नहीं, तो how would it travel? तो वो उसी जगह पे जहां bark के ऊपर जहां तक phloem है, वहां तक जाके वो deposit हो जाएगा. So if you look at this diagram here, so here what they have shown you, इन्होंने phloem को remove कर दिया, which was the bark, ठीक है, but यहाँ पे सिर्फ xylem है, ठीक है. So ये करने के बाद over a period of time, what will you see? That a bulge actually forms above the ring that you have removed here. So since case source end से definitely, because it has to go to the sink end, अब यहाँ पे it starts to accumulate. ठीक है, and if we see that this area contains very high concentration of sugar there, so this is one evidence that supports that phloem actually is involved in translocation. ठीक है, so this is one evidence that actually supports this idea. ठीक है. Secondly, what other evidences are supporting this idea is that the movement of organic substances which takes place in the plant could be tracked using a radioactive tracer. So radioactive tracers can be generally like they use these carbons. So again, they can use these. Then aphids and insects, they could be used to investigate the pressure in the phloem. So what do aphids do? Okay, if they're actually like they're feeding on the phloem. Okay, so or then actually if we see the fluid that is collected from the aphid and you analyze that, it contains many sugars. Okay, so this is also like just to see how phloem is actually involved in translocation. Right, so it is collecting the fluid from the phloem. Okay, and this fluid actually, if you analyze that, because see, you can see this is a sieve tube. Right, so this is one supporting evidence. Then they're saying when a metabolic inhibitor was placed inside the phloem, translocation was halted. So metabolic inhibitor, but remember as we were talking about this translocation is actually an active transport, especially जब हम Apoplast Pathway की बात कर रहे हैं. So when a metabolic inhibitor was placed there, okay, so the translocation was halted. So as we are saying it is an active transport, तो ये also like suggest कर रहा है कि since we are saying active transport is involved, so we are true in saying that. Okay, so these are a few evidences, okay, which are supporting actually the idea of this mass flow across the phloem. ठीक है, अच्छा, अब the objections जो है वो ये है कि they're saying कि sugar not just travels to the sink with the highest potential, but to many different sinks. अब sink एक ही तो नहीं है, it's not just the root. अगर आप for example you take the example of this tree, तो जरूरी तो नहीं है कि खाली रूट्स पे ही जा रहा हो, इसमें ग्रोइंग ब्रांचेस होंगी, ग्रोइंग लीव्स होंगे, ठीक है. सो वहां पे भी तो सिंक है. तो दिस इज व्हाट दे सेइंग के sugars not just travel to the sink, okay, but they go to many different sinks as well. Then a barrier would be created by the sieve plates to obstruct mass flow. ठीक है. फॉर एग्जाम्पल अगर सीव प्लेट्स के अंदर आप बैरियर्स लगा दें वहां पे, ठीक है, सो मास फ्लो ऑब्स्ट्रक्ट हो सकता है. ठीक है. सो दीज आर जस्ट एक्चुअली व्हाट दे हैव tried to look for. These are the objections. So have a look at that. So looking at the evidence, there is still a lot to research. Okay. So they have actually, like through microscopy, they have seen that there are adaptations of companion cells for active transport. If the mitochondria of the companion cells are poisoned, translocation stops. So these are a few very selective, the supportive evidence that I've seen. The flow of sugars in the phloem is about 10,000 times faster than it would be by diffusion alone, suggesting an active process is also involved. Aphids can be used to demonstrate the translocation of organic solids in the phloem. Using evidence from the aphid studies, it has been shown that there's a positive pressure in the phloem that forces the sap out through the stylet. The pressure and therefore the flow rate in the phloem is lower, closer to the sink than it is near the source. Okay, the concentration of sucrose in the phloem sap is also higher near to the source than near the sink. So again, aphids में कि अगर वो sink end पे कर रहे होगे तो definitely concentration of phloem sap would be less there. ठीक है, definitely अगर source end की तरफ है तो you can definitely analyze. ठीक है, so जो वही objections की जब हम बात करते हैं, some questions remain. Not all solids in the phloem move at the same rate. On the other hand, sucrose always seems to move at the same rate regardless of the concentration of the sink. And no one is yet completely sure about the role of the sieve plates in the process. Right? So these are again a few evidences. So with that, we have covered the process of translocation as well. But I would like you to actually have a look at an animated video where they're showing you both the process of transpiration and translocation. And then we'll move on to the next topic.
In order for a tree to carry out photosynthesis and maintain its overall health, water and nutrients need to move throughout the entire tree, even against gravity. How does a plant manage this without an organ like a heart that pumps fluids? As we will see, water is pulled from the roots to the leaves through a process called transpiration. In addition, water potential drives the movement of water from one area of the plant to another using osmosis, gravity, and the surface tension of water. Transpiration begins in the leaves. The arrangement of cells and structures in the leaf facilitate the movement of gases and water into and out of the leaf. A leaf contains several layers of specialized cells. The upper epidermis is one cell layer thick and provides a protective covering. Below that layer is mesophyll tissue. Cells in the palisade mesophyll layer are sites for photosynthesis and secretion, as well as storage of food and water. The spongy mesophyll layer contains a looser arrangement of cells, where spaces between cells aid in gas exchange and the passage of water vapor from the leaves. Throughout the lower epidermal tissue are stomata, which are microscopic openings flanked by guard cells. Gases pass into and out of the leaf through these openings, as well as water vapor evaporating from the leaf, a process known as transpiration. The spongy mesophyll layer contains arrangements of vascular tissue, consisting of xylem and phloem that are specialized for the transport of water and nutrients throughout the plant. The vascular tissue extends from the leaves, through the stem, to the roots. Water is transported in xylem from the roots, where the water potential is higher, up to the leaves where the water potential is lower. The arrangement of the tissues, the functions of the cells, and water potential determine the direction in which water will move through a plant. Water passes out of the leaf as water vapor through the stomata. The water vapor lost from the leaves is replaced with water that enters through the roots and is brought up through the stem in xylem. Xylem is composed of vessels, which are continuous tubes formed from dead, hollow, cylindrical cells arranged end to end, and tracheids, which are dead cells that taper as the ends overlap. This arrangement, and the polar nature of water molecules allow water to pass in an unbroken stream through the xylem, from the roots up through the shoot and into the leaves. Adhesion is the attraction of water molecules to a surface, such as the wall of the xylem. Cohesion is hydrogen bonding between water molecules. Together, adhesion and cohesion allow water to move through the xylem in a continuous stream, from the roots up through the stem to replace water lost from the leaves through the stomata. Water enters the plant through the epidermal cells of the roots and travels into the xylem. Water potential in the cells of the roots increases when symporter pumps in the plasma membrane allow protons to pass into the cell, traveling down their concentration gradient. These pumps couple the transport of protons with the transport of minerals and other solutes into the cell. Water follows into the cell, driven by osmosis. The presence of aquaporin channels in the membrane enhances osmosis, allowing bulk flow of water from the soil into the roots. The other main vascular tissue is phloem. Phloem transports carbohydrates and amino acids that are produced in the leaves to cells in the roots and stems where they are used and stored. Conduction in phloem is carried out through two kinds of elongated cells, sieve cells and sieve tube members. Most angiosperms contain sieve tube members. Both types of cells have clusters of pores known as sieve areas that are abundant on the overlapping ends of cells. These structures aid in the movement of carbohydrates, like sucrose, that are manufactured in the leaves and carried in the phloem throughout the plant, a process called translocation. Turgor pressure increases in the sieve tube members as sucrose from surrounding cells is brought into phloem through active transport. Water then enters phloem from xylem by osmosis, which drives the transport of carbohydrates in the phloem. Water movement in vessels is one way, while transport in sieve tube members can go in both directions. Water potential is an important driver in both xylem and phloem transport, but only phloem transport utilizes both active and passive transport. Our heart pumps blood throughout our bodies to provide nutrients and water to our cells. Vascular plants can accomplish this same feat without a heart, using transpiration, water potential, and translocation to move water, nutrients, and minerals to all cells of the plant.
So this is the last part of this chapter where we have to exclusively talk about xerophytes mainly, but it's better that you also know about other type of plants. So mesophytes are the plants which are able to take up sufficient water to replace transpiration, right? So this is most of the plants actually are mesophytes, right? Hydrophytes are the plants which live either partially or completely submerged in water, right? And these here actually there is problem with oxygen uptake. While xerophytes live in areas where water loss via transpiration is greater than water which is actually taken up by the roots, right? So here we are particularly talking about the xerophytes. So xerophytes are generally known as dry, because xero means dry and phytes definitely for plants. So these are the plants with the structural and physiological adaptations that enable them to survive in hot and dry conditions. So when water is abundant, the rate of transpiration is about the same as any other plant. But during drought and prolonged cases where actually there is a shortage of water, so they definitely have some adaptations which make them successful, right? So generally, if we talk about plants, plants are designed to prevent water loss. Because we know that they have these waxy cuticle. They would try to prevent water loss, right? So they would have waxy cuticle. They would have stomata on the underside of the leaf. Definitely there's a reason. Agar upar hi surface pe honge, to more water would be evaporated, right? And then stomata, they remain closed at night. So they are designed to prevent water loss. Or deciduous plants, they lose leaves in the winter. So leaves nahi hai, so you can think about stomata nahi hai, so definitely water loss comes. So these are different like mechanisms that plants actually, or adaptations that they have to prevent water loss. So they have waxy cuticles, the stomata on the underside of the leaf. They remain closed at night, the stomata. Then deciduous plants lose leaves in the winter. All right, then. Let's talk about, since we're talking about these adaptations. So the first that we'll talk about is the reduced surface area to volume ratio, right? So the plants, since we're talking about xerophytes right now, so these plants will have thick leaves rather than thin, broad leaves. Okay, so thick leaves, so you can imagine कि ठीक है, surface area definitely thinner leaves का surface area ज़्यादा होगा. So they will have thick leaves rather than thin, broad leaves. Some will have modified leaves which are spines. Okay, they also protect the plant from small mammals which would otherwise remove the water from the plant. So again, some adaptations by these xerophytes. Thick leaves rather than thin leaves. Some leaves are modified into spines. Okay, and they protect the plant from mammals because they would remove water from the plant. Right? Now, when we talk about the thick waxy cuticle, so this waxy layer that covers the epidermis, it may be thick to ensure that virtually no water can escape through it. Right? Some plants also have thicker epidermis. Okay? So it's this waxy cuticle layer. Okay? It covers the epidermis. Then the epidermis itself can be thicker as well. Right? Then talking about the hair and the spike. So what these do, these hair and spike, they trap a layer of still air between the hair. Okay? Which increases the thickness of the uppermost layer. ठीक है, because this uppermost layer which is epidermis, ठीक है, अब उसके ऊपर hair और spike है, they are actually going to trap air as well. अब epidermis itself is one layer और उसके ऊपर एक और layer बना देगा air के which acts as insulator then, ठीक है. So it increases the thickness of the uppermost layer, कि पूरी proper thickness बढ़ जाएगी फिर epidermis itself and then the air, the layer of air that is above it. So this reduces the water vapor potential gradient. All right then, they also have sunken stomata. Okay, so if you look at here, stomata at the base of the pit. Okay, so they are sunken. The stomata sunken in pits creates local humidity. Okay, it creates a local humidity. It decreases exposure to air contents and decreases the water loss. So in these plants, okay, for example, we'll discuss the example of Maram Grass. So they have these sunken stomata there. Then water storage, parts of these plants may be able to store water, such as the stems would be swollen, right, of cactus. Okay? So swollen stem of the cactus or the thick fleshy leaves of succulents. So they have these fleshy stems again because they would definitely be storing more water. Here actually you can see Maram Grass, right? So this is a photo microgram. So you can see, you can actually have a look at these hair there. Okay? So what are these doing? They would actually trap the water vapor, reducing the water potential gradient between the leaf and the air. Okay, then you can look at the outer epidermis. So it has a small but thick walled cells. Okay, there's a thick cuticle. Okay, और जो outer epidermis में there is no stomata. So again, adaptations to conserve water. So thick walled cells, thick cuticle. Here we talked about okay, what would the hair do? They would trap the water vapor here in this case, reducing the water potential gradient between the leaf and the air, right? Then this hinge, these hinge cells here that you're looking at, they lose their turgidity and they collapse in dry conditions, causing the leaf to roll up. So the leaves here in this case also rolled leaves. Okay, so you are not to use the term folded leaves. आपको जो term use करें, that is the leaves are rolled. Okay, so leaves rolled up because of this. They lose their turgidity and they collapse in dry conditions. And then if you look at the stomata, so these are in pits to trap the air with moisture close to the openings. So we have sunken stomata here. Because I have a close look at the diagram, understand it. This is another diagram that you can see. This is a light micrograph of transfer section of a rolled Maram grass leaf. So here you can look at, okay? So they've shown you the rolled leaf here. Okay? Then leafy hair, sunken stomata, and a waxy cuticle. So you can see. Okay, these are the leafy hair that you can see, right? All right then. So just quickly, let's go through the list of xerophytic adaptations. We've already gone through them one by one. But what is this thick waxy cuticle doing? A lot of water is lost through the cuticle. But if it is thick, it minimizes the water loss, and it is commonly found in evergreen trees and holly. Sunken Stomata, this reduces the air movement, creating a layer of moist air, reducing transpiration, right? Reduced number of stomata, reduced transpiration and gas exchange capabilities, reduced leaves. So we saw कि वो spines की form में होंगे, ठीक है, या thin needle-like. So this reduces the surface area to volume ratio and water loss. Conifer leaves are thin needles. Then hairy leaves, the hair create a layer of moist air, reducing transpiration. Maram grass have these hairs. Curled leaves or rolled leaves, right? Conifers, stomata in an environment of humid air, reducing transpiration and water loss greatly. Succulents, they store water in specialized parenchyma tissue when water is in plentiful supply. For when it is not, cacti and aloes are example of succulents. Then leaf loss, losing leaves in long dry seasons allow for less water loss through transpiration. Right, so you can again like just add these to your notes. Various adaptations by xerophytic cells to conserve water. And then you can have a look at this, right? So you can see actually the succulent stems, so they will store water. Right, here you can look at cactus, so you can see flattened leaves, very thick leaves here, okay? And that's about it then.